All right. Well, I want to thank you so much, Sue, for having me. And this has been a really terrific meeting. I feel like I'm learning a lot. So thank you all for being here. I wanted to talk a little bit about the project that I've been working on, funded by my HHMI grant. And it really started in 2014 with the development of the HHMI Pathways to Success program. And it's a multi-tiered approach to student persistence. And the motivation behind the project really came from a long-standing interest of mine in how do we retain students in STEM. And I think the need is really emphasized statistically when one looks at the national data about the student, and we've already heard reference to this, about the number of students who start with degrees in STEM who graduate in STEM. And so, but then when one looks at what's happening with underrepresented minority students, then it becomes really pretty stark. And if we look at, and these are students who, they don't leave the university, they just leave STEM. And the intention is no different, but the outcome is quite different. And so even if we look at, for example, the STEM degree attainment at UCLA, where our statistics on the surface look better in general, we still have a pretty dramatic disparity between underrepresented minority students and majority students. And so I think the easy place to go is that, oh, well, maybe the students just aren't prepared and they're coming in with a deficit, but, and these numbers are actually modified now, they're higher. The incoming GPA between the UCLA students from underrepresented groups and majority students are relatively insignificant, the differences. And in fact, what I found pretty striking as well in talking to students, and then you talk to the students who do persist, the ones who stay in STEM, and the conversation, particularly with a group of African American students that I've worked with, is that they feel disconnected as though the system is set up for them to fail. So even though they make it through STEM, they're not doing it in a way that we're eventually going to see them in the professoriate. So they're kind of persisting instead of thriving. And so one of the questions, of course, is how do we affect STEM outcomes for students? And I found this series of studies by Mika Estrada, which really looked at STEM success for underrepresented minority students. And what was striking is that skills and abilities themselves are not strong predictors of success. But what is a good predictor of success is student scientific identification. Specifically, the more a student is able to identify as a scientist and see themselves as a scientist, the more likely they are to adopt the norms of a scientist, including the kind of activities that would ensure success. And so I think one of the aspects of this that I think is also important to recognize is that even when students are staying in STEM, there may be times when they need to have sort of the resources, the resilience in order to thrive in an environment that is sometimes alienating. And so what kind of activities are associated with that? Students who have not acquired an identity as a scientist tend not to maintain the level of resilience that's needed to persist in the long term. And so this is sort of described, I think, very nicely in a study published last year that really describes that the ability to sort of move through difficult times and difficult situations is more likely to happen if the student identifies as a scientist. So what this led to sort of the question of how do we start to support student self-efficacy and scientific identification. And so from that came the program that I'm describing today, the HHMI Pathways to Success program. And the target group of this program is underrepresented minority students, or undergraduates in general, with really trying to focus in on underrepresented minority students. About 20 students a year are in the program. I'm going to come back to that number in a moment. But so in year one, there's really multiple levels of the program. First is to really develop learning communities where students begin to see themselves as part of a community of scholars. And this happens by focusing in on the part of their undergraduate curriculum where we find we're losing a lot of our students in the introductory chemistry courses. So they have workshops in chemistry 14A, B, and C. That's our intro series. And I'm not going to talk a lot about this because we piloted it last year and the students liked it and they seem to do well, but I don't have the data that I'd like yet. But preliminarily it looks like it's working. I'm very excited also about a development of a really serious mentoring program. And I think it was alluded to earlier today that one of the strongest effectors of student success is a sense that they're part of a community where there's strong mentoring. And so right now, in the mentoring part of the program, there are undergraduates, graduate students, postdocs, and I think there's very nice data that indicates that having hierarchical mentoring as well as vertical mentoring across, levels is particularly effective. And so the students who are in the program also then become mentors. So the way the students are selected for the program is first, I go to all the orientations and talk about the sort of things that we offer, but then I identify sort of the top 40% or so of underrepresented minority students and invite them to apply to the program. And so the part of the program I'm going to really focus on today is this inquiry-based research experience that happens with freshmen in their first year. And that is a particularly, I think, a central part of the program. And so that has led to the development of the Collaborative Undergraduate Research Lab. It's an honors level lab because it's quite a bit of work, as the students find out. But the students who become part of the program come in with the knowledge that they will take this in their freshman year. So it's an introduction to original research, reading papers, introduction to grant writing, giving presentations, and then the students do not need to have prerequisites. They do not have to have had AP Biology, which means that they have an introduction to all of the key material they're going to need for the lab. So I'm going to talk in a little bit more detail about this lab course, but I think the take-home message that I really want to emphasize is that this can be done with freshmen without prerequisites when they come on campus. And so I'd like to also talk a little bit about what happens after that. The students are placed in labs and they continue as part of HHMI pathway cohort activities. And what's particularly been nice about this is it's been allowed us to really interface with some of the other programs that are taking place on campus. So for example, hopefully you'll hear a little bit more about it today, the biomedical research minor, which is also funded by HHMI, Professor Utpal Banerjee, where students, after they participate in these the lab portion of the course, become hopefully engaged enough to participate in the biomedical research minor, which is really a research training activity at UCLA, as well as other programs like the MARC program and other research-based programs. But one of the questions that always comes up is how both scale and impact. And I've really thought a lot about this, and we've really focused on the top 40% of underrepresented minority students for several really key reasons. First of all, there are a number of programs that are available for students who may be struggling a little bit more, but we actually often sort of ignore those top students. But when we look at the data, they are leaving STEM at a disproportionately high rate, despite the fact that they look like they don't need the help on paper. It also turns out that many of these students go on to be kind of the key leaders in their communities on campus. They're the students who start student organizations, they start the tutoring networks, and they're also the students that people look to when they're succeeding as inspiration. And so really focusing in on these students seemed like at least a way to start practicing some of these ideas and seeing what works. And we also, I think, are thinking about this as a way of really testing some of the parameters of this type of structure to see how scalable and exportable it is. And hopefully I can convince you that there are a number of aspects of it that can be to scale to a much larger number of students. So I'm going to tell you a little bit more about the Collaborative Undergraduate Research Lab. In the time that I've had the HHMI funding, it's really been for the primary focus of my work. And this was, I think, one of the things that we heard a reference to vision and change. And I think when you listen to what students say they want, I think it's a really good indication of what we should be doing. And so I'm not going to go through all of this, but essentially what students describe is that they want a different kind of lab experience. One that doesn't have canned experimental procedures where they're actually doing something creative and they feel like they're generating knowledge. And so the Collaborative Undergraduate Research Lab is, I think, an attempt to do that. So the components of it are the following. It's a 6-hour-a-week lab. The students know the goal of the lab, and they know that part of the goal is to generate results that are publishable and interesting to the scientific community. There is a didactic component, which is necessary because we don't want to, the students don't have to have had a specific set of prerequisites, but we also include faculty guest lectures, reading and analyzing primary literature, and what we call quizzes. And in other classes, they'd be called midterms, but I find the students are a little less stressed when we call them quizzes. And they attend carefully selected seminars, which we prepare the students for ahead. When I say we, I'll tell you who that is. And then they have a 5-page NIH-style grant proposal. And part of the reason is that we talk about, what does it mean to be a scientist? So the goal in the course is to do the things that scientists do. And so when we come in, I come in and I show them a kind of a copy of a grant that I've written as a way of sort of thinking about what does it take to convince someone that the work you're doing is worth funding. And then finally, they give an oral presentation. And so I lay this out primarily to show that the main components of the course are really meant to mimic what a scientist would be expected to do. And so this is possible because I've been able to connect the project to the research in my lab. So I'm not going to go into a lot of detail, but I do need to just kind of give a sense of what we're interested in so you can see what the lab is all about. So my lab studies RNA splicing, and I think there's probably only one other person in the room who cares about the details of this. But we go from the question of how does genetic information get expressed, how is expression regulated, and then introduce them to this machine, the spliceosome. And we talk about it as being something that's dynamic, that's highly conserved, that requires energy to undergo these dynamic rearrangements, but that's important because that's directly connected to proper gene expression and human disease. So the beginning of the class is really convincing students that understanding molecular details have important implications. And so how we can study that is we can use a model organism. In this case, we use yeast and yeast genetics. And so we've developed a genetic screen in which there's a mutation in a splicing factor that my lab is interested in. And then they perform a transposon insertion mutagenesis screen to look for suppressors of a temperature-sensitive phenotype in the yeast. So they screen for suppressors, they identify the mutants by PCR and then whole genome sequencing. And so the particular gene, protein and mutation that we focused on is called PRP2-1, details of which are clearly not going to be important for this. But what I want to emphasize is that the students start off with very little background information about this general topic, which means that in each of the steps of the 10-week lab course, we have to present the content. And so what was the first thing I learned from this is that actually you can teach some pretty sophisticated topics within the context of the experimental procedures, and the students actually retained it. So for example, discussions of model organisms and model systems happened very early as we started kind of understanding how to work with yeast. They started to understand the classic experiments in molecular biology, gene regulation in great detail. They had an introduction to genetics, in particular yeast genetics. When we talked about PCR, they couldn't understand that without really understanding DNA replication. And in order to understand the sequencing, they had to understand sequencing in general and some things about RNA biology. And then at the end, they got an introduction to genomics when we were looking for insertion sites. And so one of the sort of eye-openers for me is when I taught upper division molecular biology. These are juniors and seniors. We covered the same general topics. And so when I gave the students in this class the same quiz that I gave my upper division students, the students in the freshman class did much better because it was framed within a context of a specific set of experimental questions. And so what kind of results do we look for? So what the students do is they identify suppressors, they name their suppressors, so I've called them groups, but now they've given each of their suppressors specific names. Sometimes they get very creative with those names. And then they identify validate that they have insertions, They learn how to set up controls, how to understand what a positive control is, what a negative control is. They isolate RNA and then they analyze splicing so they can see in one case, here's an example of a splicing defect, and their suppressors, the decrease in this upper band and increase in the lower band indicates that we're identifying suppressors. And so then, of course, the question is, what are the suppressors? So this gets us to about halfway through the quarter. And then the students have to write a grant proposal. So since we don't know the answer, that requires that they do research, that they analyze the literature, and they work closely with the TA in the class to think about mechanisms that could explain their results. And so this is just a figure from one of the students. This is a particularly impressive student, frankly, who developed a model based on the literature that a protein that has previously been described biochemically may be disrupted in a way that would bypass the requirement for PRP2. And so she put together this very nice model, and this is her figure legend. And so at the point that she started this project, of course, she knew nothing really about splicing, but my sense was that the enthusiasm of realizing that you might be discovering something that no one else has discovered, I think really spurred some exciting work. And so I have to highlight the work of a really talented postdoc in my lab, Stephen Douglas, who developed a script called Find Insertion Site that allowed us to identify the sites of insertion. So I'm not going to go into the scientific details, but we found a number of things that looked interesting that the students identified. But once they found the insertion site, the next step was for them to figure out what it could possibly mean. And so the last 2 weeks of the quarter were essentially the students working together to think about mechanisms that could explain their results. And so this is an example of the final presentation. These are the groups. This is the entire group, along with the academic coordinator, who I'll tell you a little bit more about, Azad, as well as the TA for the class, Sam. And so after the class ended, a couple of the students spent the summer focusing in more on their particular mutants, working with their graduate mentor, Samantha, who was a class TA, and it turns out also her research project, she switched to studying PRP2. And so, but the big question is, okay, what is the impact of this kind of activity? So the first thing we did at the beginning of the quarter is we give the students a pre-test of basic concepts, not concepts like, okay, what part of the spliceosome does this, because obviously nobody's going to get that. But what we ask them are things that they should know. They're freshmen, they've gone through at least 1/4 of chemistry, so we ask them basic questions about calculations. A question that many of them, if they've had any biology, they should know, well, what is transcription? And so this is just to illustrate that even though we were not focusing in on these in the lab, these topics, that you see both they do better and there's less of a spread between the students. And we've seen this the first year we did it, the lab, and the second year. But we also ask questions about their sense of ownership of the material, as well as their self-perceptions. So for example, we ask the question, I understand what it's like to do research in a laboratory. And what's shown in blue is their pre-test answers, and the red, their post-test, post-course answers. And so what's exciting is most of the students are either in labs or in the process of being placed in labs. But the striking thing is that, for example, if you look at particular students, there's a pretty dramatic increase in their sense that they know what it's like to be in a lab. These two students are highlighted, and a couple of others who are not highlighted because they've now been accepted into the biomedical research minor, where they're really focused in on research development. And so a quote, for example, one of the students, which I really like, is who said that I learned that doing research is more than just thinking about a specific problem, but looking at the experiment's connection to the rest of a mechanism or organism, which for a freshman I think is pretty sophisticated to be able to put a set of experiments into context. The question, I consider myself a scientist, so this was the first year we ran the lab. And you can see, there are certain students who came in with a fairly low perception of themselves as scientists, which improved dramatically by the end of the quarter. This student went on and found her own research opportunities in her hometown at Davis. But even the students who came in, pretty confident, if you read kind of what this particular student says, it's clear that that even thinking of themselves as scientists, there's really little knowledge of either the subject or what that means. But that after the class introduction to research changes that. And I think the second year, the results we're pleased to see are I think, even bigger differences. But this one, I think, quote from one student, I learned more of what it means to be a scientist and that I love it. So the students were also asked pre and post tests what their career aspirations are. And this student in particular is interesting because their original career aspiration was MD. And I think at the end of it, it switched to MD, PhD. Not that that is necessarily what the student's going to do, but just the sense that a PhD is possible has changed. And again, I'm highlighting some of the students who are in the minor. And I do that because I want to emphasize that there is a synergy between the activities on the campus. They start in their first couple years here, but they really can integrate into other types of research environments at UCLA. I'm not going to go through all of this, but the question we asked was to describe how their experience in the class differs from other classes. They talk about impact on career goals. They talk about feeling like participants and that their contributions were actual contributions, and that the experience was as powerful because they found themselves engaged not only with the professor and teaching assistants, but also fellow classmates. And so the students talked about going back to their dorms and feeling like nobody else really understood what they were talking about because they were like trying to figure out what PRP2 did. It's like splicing. So, so some of the lessons, I think, first of all, I always put this here because there's always this sense that you cannot work with RNA, and we never had RNA degradation in the lab. That there are really unexpected science superstars that can really get charged by thinking about science in more experimental contexts. That they can learn very sophisticated concepts when it's taught within a relevant context. That model organisms are fun. Freshmen actually really like to publish. They're still asking me when the paper will come out. And experiments failing can actually be really great moments of opportunity. I can tell you about plenty of those during lunch, but my first response was often panic. The students' response was excitement that something had happened that we didn't really know what the answer was going to be. It's important for students to see what community feels like, and what I mean by this is that when students go to a seminar, they see that scientists interact with each other, and people are friends, and that they know that science is a community and that they could be part of it. And I think this idea of impact, that the only way to really change the conversation about science education is changing in a class of 300 students. I think that what I'm beginning to think is that if you find some really key ambassadors and do the right thing in the classroom, that you can have a real multiplier effect. And then in the case that something I've really learned is that the members of my lab, as long as it's clear what the benefits can really be partners in this. And so, these are just some quotes about the mentoring program that I don't really have time to go into, but we're in the process of assessing the effects of the mentoring aspect of the HHMI Pathways program. And one of the things that has been the most striking is that the mentors have been instrumental in helping the students develop a plan for getting into research, particularly the graduate student mentors for the undergraduates. So some of the future directions are the following. We're really hoping to expand what we're doing to open more choral sections. We really want to focus on transfer students. Part of the problem is that transfer students we find are not having research opportunities early because they come in and they've got 2 years. But if we can give them a research experience within a lab setting, that could maybe shift the dial for those students. to expand some of these questions beyond splicing, although I love splicing, maybe not everybody does, but we've been thinking about how to partner with colleagues who might be able to, might be interested in developing other types of laboratory experiences, maybe using yeast as a model system. Intercampus collaboration, and this is specifically because one of my former postdocs is now a faculty member at Mount St. Mary, and so we're doing the lab in parallel and are hoping to be able to bring the students together to talk about their results from both campuses. The key things with the overall program is assessment, assessment, assessment. Right now, we're trying to assess how well the workshops are working, and one of the challenges is really disaggregating positive effects that may be from the workshops, from the mentoring, and from the collaborative undergraduate research lab. So the most controlled experiment would be to leave one of those out, but frankly, I think it's probably irresponsible to do it that way. And to really expand the training for mentors and workshop leaders, one of the real surprises has been that because we're creating a community, it's beyond just the 100 or so students who will go through the program over the course of the grant, is that it's really helping to develop a culture around mentorship and teaching and engagement with students, and a recognition that there's talent everywhere. So I'd like to acknowledge, this is Azad. He's an academic coordinator and a research scientist who works with me in my lab, but he has been amazing in helping to develop the lab and is just wonderful to work with, and he's great with the students as well. We've had great assistance from colleagues who have a lot more experience developing labs at UCLA, Chris Reddy and Jordan Moberg. Srivat's and Sam are both graduate students in my lab. Srivat's TA'd the first year, Sam TA'd this year, and they've also helped to develop the lab. Both of them, I think, have, at least anecdotally, indicated a stronger interest in teaching, I think, after having had to do a lot of the groundwork for helping to launch this. And Stephen Douglas, who's a postdoctoral fellow in bioinformatics and who I think is going on the job market next year. So these are the amazing students who have been in the cohort so far at our end of the year party. In addition to being very grateful for funding by HHMI, I've been really lucky to be surrounded by great colleagues. My dean, Victoria Sork, has made commitments to institutionalizing the kind of things that we're doing in the lab. My chair, Utpal Banerjee, who has been helpful in so many ways, but one of the really key things he's done, which you've already heard allusion to, is that he's illustrated that you can be an amazing scientist and you can do great teaching and you can care about both and be successful. And he's created a culture like that in our department, which I'm very grateful for. Ira, who's the director of the biomedical research minor, who helps to place these students in labs and is an endless source of advice. A program coordinator, Ina Thorner, academic advising, and Evan, who kind of helped me think through whether the screen would actually work. And thank you for having me.
Wanted to know how you recruit students for this program, if they're intimidated by the 6 hours, what quarter do you have this in? Is it kind of word of mouth, and how you do that?
Yeah, it's a great question. So the first year, when we were just barely getting it running, I ran around to orientations describing the lab and then recruited students just by figuring out which students were coming to UCLA and looked like they were interested in science degrees. And so that's how we filled the first cohort. I think cut to now, which is that we've had probably almost 4 times as many applicants as we have slots. But students are totally excited about the research part of it, because I would have thought it may be intimidating to come in, but I think setting the tone that you don't have to already know how to do research, but that we want you to be part of a research project, students seem to be actually really excited about it. So the recruitment process is to both kind of talk in forums where incoming students are going to be, but also to send invitations. I send invitations directly to like the top 40% of the underrepresented minority students who get accepted to UCLA. And the dean has just started a scholarship to kind of attract students from particular schools who might be good candidates for UCLA, and part of their scholarship package is that they can participate in the program. And that was the difference between them coming to UCLA and going other places. So I think that research opportunities are a total draw, even if they're a lot of work. I offered it in the spring. This year we're doing it in the winter. Hopefully that works.
I'm sorry, I have one more question. So do you ever get students that are not underrepresented minorities saying, well, hey, I want a piece of this, I want to do this too?
And I have taken all of the non-underrepresented minority students who want to be in the lab, I've always taken them into the lab. So, and the goal is to scale this kind of activity, maybe not exactly this, but this kind of activity in a way that a lot more students can participate, but yeah.
Does this waive a requirement for another lab course?
Yeah, so I meant to mention that. So it can fulfill one of the requirements for the biomedical research minor. And that's it. But the thing that anecdotally what students say is that it's very difficult to get into a lab at UCLA, at Berkeley. It's difficult to get into labs. And what students are telling each other is that their chances of getting into a lab are higher if they've taken a course like this.
Thank you for this really inspiring talk. It looks like the students are making huge gains in their identification with being a scientist. So there is a research coming from educational psychologists such as Jesse Smith from Montana State University and our own Dustin Thoman from SDSU, San Diego. Talking about the utility value, so how important it is for URM and first generation students to be able to explain how the research they're doing or learning about relates to their communities or their families. Have you seen this, and have you been addressing this?
I think that's I was really struck by that literature as well. Something that happens in almost all of the grant proposals is the students always find a human health element. They almost always find something that connects to the possibility that what they're doing could have a broader impact on society or on communities. It was interesting, one of the students found a paper I'd never seen that described a developmental disorder that appears to be prominent among Mexican Americans. And one of the common mutations seems to be in something that she thinks may be connected with PRP2. So it was literature I'd never seen, but I think the inspiration comes from different places for different people. Penny?
Can you hear me? I'm Yu Pakapo from UC San Diego. I'm wondering, from the first cohort that you had, when they went back to the traditional class, what did they think?
When they went back to a
Traditional science class or lecture and lab, what did they think of it?
Well, so one of the things that is really interesting is one of the things they came back and said repeatedly is that their life science courses, which they often weren't getting until later, that they had had a taste of the biology early enough so that when they were doing some of the other things that felt like prerequisites, that they felt less like barriers. Does that mean that they were courses that they were like, okay, I can get through the physics because I know what biology looks like. And then they also said that, no offense to physics, because physics is a wonderful topic too. But the other thing is they tended to feel like they did better in their life science core classes, which may not be surprising. They've kind of done PCR, so learning about PCR feels a lot more tractable. And from what I can gather, they did very well in those classes where they'd already had lab material related to those classes. And so they communicate that as well. So I don't think it is a negative impact on their perception of their other classes. I think it helps.
Yeah, I just wanted to add that I've seen the same thing that you have with freshmen, and in a couple of projects like this that I've run with students, they are the prerequisite structure and the need to fill out check boxes to get a major really makes them passive about being able to learn something on their own. And so I've had students say, well, I can't do that. I haven't had a course in that yet, right? And so doing it this way, the way you're doing it, makes them realize they actually don't need a course to do something. And if they carry that through the rest of their it's so powerful for them.
They learn how to find answers when they need them. And they learn how to be critical of things that they find online, because it's not syncing with what they've heard us talk about in lecture. So it's been interesting to see how people use publicly available data to learn. And I think that's something we don't teach students enough, is how to be critical of that. But when they are trying to figure out things because they haven't had prerequisites, I think it's really empowering.
So, okay.